Everybody, thanks for coming. It's my pleasure to introduce our speaker today, Catalina Cuellar Gimple, who is currently at Humboldt State. She's an associate professor there, tenured, yay. But she has an interesting sort of history and has sort of toured all kinds of different places for her career. She was born and grew up in Colombia, where she went to her undergraduate there and got interested in environmental education and some in microbial. Her first interest in microbial biology were sort of there. She did her PhD at University of Texas in Austin, where she spent some time at the UT Marine Lab at Port Aransas. So she has a history with marine labs. So she's getting some flashbacks today, positive ones, I hope. Then later on went and did a postdoc at Florida State University. And I think I would sort of characterize her work, as she's going to tell you about today. She's an ecologist who sort of blends, I would say, ecological theory, microbiology, natural history into sort of being able to ask questions about community ecology. And this is sort of what I really appreciate about her, is she's sort of, I feel like, equally at home in the forest, in the ocean, and in the lab in some ways. Maybe you don't like all those three the same, but your work easily crosses all of those areas. And she's worked on crabs, sea anemones, marshes, and pitcher plants, which I think is mostly what you're going to talk about today, as her work on pitcher plants and negative biodiversity function relationships in microbial community assemblies. So please, let's welcome Catalina to Bodega and take it away. Yeah.
Thank you. Does this sound good? Everybody can hear me? Thank you, Jay. That was a wonderful introduction. And you did a wonderful job at letting everybody know that my heart really lays in the ocean. I think that's where I would rather be. I like the forest and the mountains too, but the ocean is beautiful. But I'm going to talk a little bit about our work in pitcher plant, the pitcher plant system, focusing on that biodiversity ecosystem function relationship. And I was going to do, let's see if this works. I was going to tell you again that my heart lies in the ocean, and that a lot of my work early on was on crustacean associated microbial communities, and then I took a detour with pitcher plants, and I have a lot of work pending that we're hoping to get funded that allows us to work in constructed marshes in the base up there in Humboldt, thinking about nitrogen cycling, and hopefully gets funded. But really, like, a lot of these make me, and reflecting with you all today, make me realize that all of these systems are interesting to me because it allows me to ask the questions that I'm interested in. And each of these systems allows you to think about the same questions a little bit differently. And so I want to start my talk by going back to this general idea that has been in ecology and central to ecology for many, many years, probably 3 or 4 decades, depending how you start counting, and it's the relationship between biodiversity and ecosystem function relationship. There is a large amount of evidence that supports the idea that as you increase the number of species, particularly this evidence comes from plant communities, you increase the number of species, you increase the function. And function, we're talking about primary productivity and carbon accumulation, biomass, and things like that. And the opposite is also true. If you're losing these species, you're going to lose function. And so these patterns that repeatedly come up became a basis for an urgent call for conservation in a time of habitat destruction and climate change, and you know what's happening. I would argue that perhaps equally important and urgent, or even maybe more urgent, is for us to understand the relationship between microorganisms and their functions. And there's many reasons for this, but I want to tell you 2 that I think are critical. One is that microorganisms are the driving forces behind the biochemical cycles in our planet. Most of the transformation of the nitrogen cycle, about 50% of the carbon cycle, and then you have sulfur and silica and magnesium and iron, all of these elements that depend on microbial processes to move around on Earth and make sure life can happen. In addition, there's an enormous amount of evidence showing the role in the health and well-being of animals and plants. So in the context of host survival and reproduction, they're playing important roles. And so if we want to take care of ourselves and we want to take care of our loved ones and of any organism, plant or animal on Earth, and if we want to take care of our planet, I would argue that we need to know what these guys are doing. And these are my cartoons of bacteria. They're really expressive. So because we were interested in this when I started at Humboldt, we took a deep dive into the literature thinking, okay, what are the mechanisms that drive the relationship between biodiversity and function, and how do they apply to microbial organisms? And some of the things that we found were 3 general mechanisms in the literature that explain these patterns. The first one is complementarity, and the idea is that when species come together, they're able to utilize the resources more effectively. They're able to support each other, kind of pack their niches, and produce a better yield that they would do alone. And so this produces that positive relationship. A second mechanism is more like a randomized sample effect, where it's the positive selection effect, where the more species you have, the more likely you are to include those species that are really functional. And the third one is really a deviation of that positive relationship, but it shows that at some point when you add more species, they're functionally redundant. So you're not really increasing the function. You obtain this kind of like flat relationship. And it's kind of nice because it allows this stability of the system where, at least at high diversities, if you lose some species, you might not immediately lose function. There's a recent review paper in 2019 that seems to support at least the fact that plant communities have these positive biodiversity ecosystem function relationships, both in theory and empirical work. But then when the authors start including different systems and different functions, they start finding kind of a different landscape. Relationships that are flat, with those apparently showing functional redundancy in yellow appear, and those negative relationships appear as well. And so from this, if you don't take anything else from this talk and you decide to leave right now, I don't want to see, but what I would like you to know is that perhaps we need to consider that these relationships between biodiversity, ecosystem function can have multiple shapes, and that keeping our mind open to how we interpret this might help us better portray particularly how non-plant systems are behaving, and specifically microbial communities. Now, for the rest of the talk, I'm going to talk to you about my obsession with negative biodiversity ecosystem function relationships. I think this is an unexplored and understudied relationship that might allow us to link community dynamics with ecosystem dynamics in a very unique way. And there's 2 particular reasons why I think this. One is that negative biodiversity ecosystem function relationship really depends on the identity of the species that is providing these functions. Only the communities with lower diversities, where we can probably track these species, are gonna provide these elevated functions. So there's something about the traits, the tolerances, the ability to interact with other species or survive interactions with other species, and the functional outputs of these species that is unique and only happens at low diversities. The other reason is that the functions themselves seem to be different. At low diversities, we see functions benefited that are only appearing in species that are related to one another. So there's phylogenetic constraints to some of these. They might be ecologically, energetically expensive, so they require large enzymes and investment. And they might be associated with weak competitors. These species are investing in large enzymes, in large genomes, and perhaps not being able to grow as fast as other species that are not performing them. And this is what has been described in the literature as negative selection effect, is the species are selected against when the community increases in function. And so what I really want to do today is ask whether we can detect and characterize negative biodiversity ecosystem function relationships. And to do this, I'm going to take you on a voyage of searching for this. I want to start by going to the field and looking for these negative relationships. And then I want to go to the lab and see if we can reproduce them and manufacture them almost. And then I want to use some math to see if we can understand where they come from. You probably guessed that what I'm doing is doing a little trip between the biological realism of what's going on out there, yeah, I see some nodding, and the mechanical or the mechanistic explanation of why they might be happening. So let's start with the system. All of the story that I'm going to tell you focuses on these pitcher plants. These are carnivorous plants that produce modified leaves that look like little cups. They start sterile, and when they open, they start accumulating fluids and microorganisms, and they start producing nectar pretty early on at the tips and entrances of their leaves, which leads to attracting animals. I picture only insects, but they attract a variety of different animals that fall into the trap. They drown, die, and then they get consumed, fully digested by the microbial community that then provides nutrients, specifically nitrogen, sometimes phosphorus to the plant. These plants live in soils that are mostly devoid of or have really, really low levels of nitrogen and phosphorus. So the whole point of these carnivory strategies is to supplement their diet in terms of nitrogen and phosphorus. So this is a really nice system that includes the assembly of a microbial community, and the function that they provide is their ability to degrade a prey, which benefits the host, allowing them to access nutrients. Now I'm going to focus on, in my lab in reality, we focus most of the time on this portion, but there's a lot of people doing this other side of the story really eloquently. You should invite them to see the other side. I can give you a list. I would argue that this is an aquatic system, so we can consider it an aquatic habitat. But you can argue with me later about that. And so the first story that I want to tell you is negative biodiversity ecosystem function in the field, and this is a story of succession within pitcher plants. But let's think about succession. You know that succession has been in ecology from the very beginning, a laboratory for us to understand how different types of environmental conditions and biological interactions lead to the development and change of species composition over time. And the earlier ideas suggested that as succession proceeds, species accumulate, and as species accumulate, then you have the increase in ecosystem function until species reach a climax scenario of maximum species and maximum ecosystem function. If you plotted this in a BEF relationship, it would look like a positive relationship. And there's some examples of this happening. But now we know through modern successional theory that not all successions look like that. Some successions start with really high diversities and then drop, and some successions go up and down, and there's a vast variety of ways in which succession can happen. In fact, I've tracked differences in successional trajectories within a single crab happening in different directions. So succession can happen in many different ways, but perhaps if we can track the mechanisms that are driving changes in diversity, then we can learn what impacts diversity and function as the system changes and learn something. So what I want to do is do that on the pitcher plant. Luckily for me, Tom Miller and Casey Terhorst described the succession within Sarracenia purpurea in 2012, which feels like a million years ago. And this starts with a little rundown of the simplified trophic system that happens inside of these. And so let's say we start with prey. So these are mostly ants in the Sarracenia system in Florida. They get eaten by bacteria and mites. Bacteria are consumed by like a secondary consumer level of protozoans and rotifers, which then get consumed by the trop predator, which is a terrifying larvae of the mosquito Wyoma smithi. If these things were our size, it would be like a terror show. They're so scary. And this is a specialized mosquito that only lays its eggs in Sarracenia purpurea. Early on, when the plant opens, oh, I forgot to say something. We're gonna focus on bacteria. This is all we care about right now. You can care about the mosquito later, but right now, we're gonna focus on the bacteria because the bacteria is providing most of that degradation function. As the leaf opens, the first thing that happens is that the leaf is producing a lot of nectar, so it's attracting a lot of ants. And the mosquitoes are laying a lot. So we have that mosquitoes are up. This is over time, this is 10 weeks, 20 weeks, I believe. Mosquitoes are really, mosquito larvae are really dense early on, which means that they're eating the protozoans. And as I said, ants are highly abundant as well. And so imagine being that bacteria. Your predators are down, and your food it's a banquet. And so we would expect bacteria to be highly abundant and diverse in this scenario, but things change. And so as the leaf ages, you have no more of a position of mosquitoes and barely any ant is falling in. So what you're having here is a scenario where the intermediate consumers are diverse and abundant, and you have no food. Bacteria should be dropping in abundance and diversity. So I would argue that this is a succession driven by a shift from high nutrient availability and low predator pressure to high predation pressure and low nutrient availability. Pretty dramatic. So let's see whether succession can influence the function and diversity of pitcher plant bacteria. And particularly, I want to focus on that idea that the leaf is associated with nutrients and there's protozoan predation, and which one of these might be driving the differences. So we did a field assembly experiment. We allowed these to assemble in the field, and then we brought them to the lab for experimentation in a replicated design. We went out and tagged leaves every 2 weeks, and we ended up having leaves all the way from 2 weeks old to 24 weeks old, and then we collected all the fluid and pulled it together, filtered it to remove debris and larger protozoans. And then we took these pooled fluids from each age and made replicated microcosms. 5 of them had an added tetrahymena, which is a common predator in these systems, and 5 of them were controls. We incubated for, I don't remember, 7 days, and then we sampled a bunch of stuff. We did functional genes and 16 air for abundances. We did qPCR, and then we looked at prey weight change as a metric of function. So what we would do is grab mealworms and dry them, and then drop them in one of these tubes, and then take them out, dry them, and weigh them again. So that change in weight is a metric of how much was eaten by the bacteria. Oh, here it is. Look at that. So cute. And then we looked at bacterial microbial community composition and diversity with 16S amplicon sequencing. What I'm going to show you is this part. The rest you can ask me later or read the paper that is going to come out in like a month. Yeah. And I can't tell you what happened until I acknowledge the work of these students. Janaya, Jana, Nikita, Sophia, and Daniel went out with us, and this is in Florida summers, so that's bravery right there. All the time to tag the leaves and collect the samples, and they created some of the contraptions we needed, and they were amazing. This is what we found. This is a plot that shows bacterial diversity, richness, so ASVs is equivalent to species, and then in the x-axis we have the age, so the succession proceeding in weeks. The diversity of bacteria dropped over time, which makes sense to me because they're experiencing that shift from high nutrient to high predation. However, let's look at the function. The function changed in the opposite direction, increasing through time, mostly driven by these older leaves. At the end, there's an empty spot here because during winter they don't produce any leaves. Older leaves are really driving that. And if we plot it together in a plot like this, like a BEF plot with diversity and function, so replicating that plot we talked about in the beginning, what we get is a negative biodiversity, ecosystem function diversity, driven by the fact that early succession leaves have high diversity, low function, and late succession leaves have high function and low diversity. In these plots, you're gonna see that the dark dots are the predator treatments. We had no significant effects at all. And the size of the dots in this one is the age of the leaf. So how can we answer this question? We found that succession has an opposite effect, leading to decrease in diversity and increase in function. The decrease or decline in diversity makes sense to me because of the ecology of the system, but how is it that function is increasing? And so we inquired by looking at species relative abundances and their relationship with function and age, and what we found was first a subset of 60 species that are correlated with increased function, which suggests perhaps complementarity. This subset of species might be changing through time, but they're contributing to higher outputs. But the most interesting part is that one amongst them, Bolcoldaria, a Bolcoldaria species, is associated not only with increased function, but it increases through time. So this is the only species that clearly responds to succession by increasing through time. And when we took that ASV and matched it to the literature, we found that it's associated with taxa that have been isolated from the guts of insects and have large genomes with wide metabolic pathways. This suggests a couple of things. We can perhaps inquire where these are coming from, why are they associated with function, and why are they appearing more abundant at the end. It's not dispersal. I thought it was going to be dispersal. Maybe they're coming in at the end with rains, I don't know. But they're coming in at the beginning potentially if they're inside of the guts of the insects. Maybe they are oligotrophic. They benefit from the low nutrient environments. We know it's not the predators because they had no effect. But they have large genomes. And generally, bacteria are assumed to require more energy to reproduce when they have large genomes. Not always, but this will need some experimentation to figure out. And we're isolating this Bolcoldaria as we speak and playing with it a little bit. But it's possible that this is a poor competitor that thrives in those low diversity environments and that benefits from fewer opportunities of competition by expressing those large enzymes and enhancing function. And this would be an excellent example of that negative selection effect that rarely appears in the literature. So hopefully I showed you some evidence that this is not the only one. If you want to talk about it, we have another full data set in constructed marshes showing negative biodiversity function relationships with bacteria. But I want to shift to the lab. Let's go to the lab. Put your lab coat on and put your hair up. I had a girl catch on fire one time. Not in my class, it was in my class. My class is very safe. And so what I want to do is see if we can reproduce this. So we were talking about Sarracenia purpurea. Now I'm going to switch to Darlingtonia, which is our local California species. And when I emphasize something that maybe I didn't mention is these two species, out of all of the other pitcher plants, are really interesting to me because they produce almost no enzymes of their own. Most of their degradation, or the vast majority of it, is derived from the enzymes and activities of the microorganisms inside of them. So we went conveniently to our greenhouse where we have some of these, and we took some fluid from leaves and we performed a serial dilution, kind of replicating those dilution to extinction experiments, but we didn't go to extinction. We calculate that we went from 10 to the 13 to 10 to the 8th cells per mil based on OD and plating. And then the idea is that as you select smaller volumes and propagate them, you are reducing the number of species just out of the chance of selecting across. In addition to this, we replicated this, we did it multiple times in what we call series, with the idea that perhaps, because this is by chance, we can replicate this and then get different compositions. So in my beautiful cartoon example, we can imagine that we can obtain communities that are, have the green one or the yellow one in them. Does that make sense? And they're so cute. I should make a movie, bacteria movie. And so we generated different assembly series. We did 3 of these, and then each one of these tubes was used to inoculate 3 replicated microcosms. We incubated them for 4 weeks and feeding them to make sure that they were reaching the same abundances, because we wanted to focus on those differences in composition and that gradient in diversity. And each tube, again, we made some metrics. We looked at protozoans, abundances, then function. This time we used sterile fruit flies. We've been playing with different preys for different reasons that I'm happy to talk about. And then we looked at amplicon sequencing for the bacterial community composition, and we did some other assays that just to know what the underlying functions are and how the prey is being degraded. But I'm not going to talk about that. I'm just going to talk about these. And I can't tell you anything else until I confess that this wouldn't have been possible without the leaders. Marisa, Eric, Dan, Jose helped design this experiment. They participated in putting it together and running it with the support of some of the grad students and some of the other people in the lab. I was enormously pregnant at the time. I couldn't even hold that pipetter without making a mess. And so this is like totally, they're co-authors in the paper, and they were amazing. Especially Jose Alejandro was great, was the leader here. And this is what we found. So again, we have ASV richness here, and we have the change in weight in the y-axis. What are we looking for? Again, we're looking at this shape. And what we found, behold, is another negative BEF relationship. In this one, interestingly, the series was significant as much as the richness, and I want to show you how interesting the series, because of essentially differences in the composition, we see that the slope of this relationship changes, with particularly series number 3 having like steeper slope. And we can use this to kind of ask these questions about compositional effects and what is the identity of the species that are driving the higher functions. And what we found was, in this term was a Clostridium species that is associated with Clostridium related taxa in the soils that have a variety of organic matter degradation pathways and nitrogen cycling, sporulation, and these like drastic competitive relationships with related species, out of which they always lose. So this kind of supports the idea that these are species with large genomes that are poor competitors in their environments, but it also suggests that they might be competing directly with some other taxa. In this case, we were able to identify those, the antagonists that are negatively related to function and negatively related to the abundance of Clostridiums, particularly Herbinix, which in my head sounds like Hendrix, but I have to remember it's not Hendrix. And these are the competitive dominants that are taking over the communities and reducing function in some of these assemblies. So perhaps there's a component of historical contingency or a stochastic assembly where if these dominants get there first, can take over the communities and drop the function of the assembly. So I showed you these two. I'm telling you, negative biodiversity ecosystem function relationship happens in the field and can be done in the lab. So then why is it so rare in the literature? And more even so, how do these plants do it? They're widely distributed, they're rare, but they're doing fine. And so is it that this one species is dispersing everywhere and populating the pitcher plants over California and Oregon and Florida to Canada? Or is it that perhaps there's different species that fulfill this role in the range of the plant? Or maybe it's even more complicated. Maybe the BEF relationship changes geographically and we have to take into account biogeography. Hopefully not. And so to answer this question, this is super preliminary, so totally bear with me. We went out and we sampled pitcher plants all over the range of Darlingtonia and then the equivalent range in Sarracenia, because Sarracenia has a much wider range, but we tried to keep it the same. And ask whether we can answer this question by collecting fluid from them and replicating that dilution to extinction experiment across the board. And so we took 50 leaf from each side, we pulled it and filtered it the same way, and what I'm gonna show you is kind of aggregated results from this. Again, bear with me, I'm still learning what this means. So what I'm gonna show you is the estimate slope of the BEF. If it's higher than this line, it means it's positive. If it's lower, it's negative. And then if it's significant, it's gonna come up in a red dot, okay? And what I'm showing you here is the site, and these are Darlingtonia sites, these are Sarracenia purpurea sites, and they're organized from south to north. So this is, now I realize this should be the opposite, but what is, I mean, this is all made up, right? So south to north, south to north. Does that make sense? Okay. And here's what we found. Oh, and I'm showing you this because at first I thought there might be a signal from host, like the plants controlling it, or it's a latitudinal gradient because latitudinal gradients are everything in ecology, but they're not because there was no signal. This is what I found. Most of the relationships were flat, suggesting functional redundancy. And then we had two deviations. One is the Siskiyou site, the most northernmost site in Darlingtonia, that had a negative BEF, and Franklin Bog in Pennsylvania, was smack in the middle of that range, that had a positive BEF. So it's not host, it's not latitude. I'm gonna tell you what I think it is. And I knew this before, so I should have told you before, but I'm tricking you. There's an idea that suggests that the regional pool of species, the species that are available for colonization, in their functional redundancy might drive how these communities assemble inside a picture or inside any community. And this is how it works. If the functions are redundant in the pool, so the species that are floating around, ready to colonize a new leaf, if they all have, there's few genes and they're distributed across, they're redundant, and so they're more likely to lead to redundant local BEFs. If they're very unique, there's a lot of genes and they're distributed across the population, some species are more likely to have genes that others don't, then the communities are more predicted to be either positive or negative in their BEF locally. So let's see if this works. To do this, we went out and took from those samples, we did metagenomics. I'm gonna skip, we did a lot of things that I'm happy to talk about, but what we got out of this is the predicted taxonomic diversity and functional diversity of these pools that we use to assemble the communities. And when we plot it, it looks like this. This is the taxonomic diversity. It's not a lot of variation. Maybe Sarracenia has more variation than Darlingtonia. I'm not gonna dwell too much on that. There was no range and no latitude effect. Functional diversity, again, seems like Sarracenia has a little bit more variation, and I'm labeling here the sites that were different in the slopes. But what I really want to show you, it's not this, don't even look at this. First, because they're ugly, but second, because the point is that when you divide the genes by the taxonomy, you get the potential. So you get that metric of redundancy that is gonna appear here. Okay, and what I see is that the Pennsylvania site is the highest, suggesting it has more unique genes per taxa, while the Siskiyou site is the lowest. Not by a lot, but it's the lowest, suggesting it has more redundancy. And if this is true, it means that the availability of species and functions in the possible colonists might be influencing the potential BF relationship of local communities in this way, where low regional functional redundancy leads to positive BF, and high regional functional redundancy leads to negative BF, and it feels like a tongue twister. So don't quote me on this, we're still analyzing this data, but I'm really excited about the possibility of demonstrating at least a piece of what explains these patterns. But it also shows that negative BF is rare. Out of 9 sites, we got 1. And so we went back to the board after showing that they exist and that there might be negative selection. And we try to ask if we consider all the different mechanisms that are required to assemble these communities, what is needed, what needs to happen to produce a negative BF? And I'm really lucky to have met people that do math. I mean, I love math, but that's like another level. And so this is a fruit of a working group funded by the NSF that was awarded to Matthew Leibold and Pedro Perez Neto and Elise Devolt. We met in Montreal a few years ago to talk about statistical theory and microbial communities. And then while we were at it, I convinced a group of them to do something completely different, so don't pay attention to that. What we talked about was going back to this model that people have been discussing for a while. It was published by Leibold et al. In 2016, and this is the whole proposal of you have some species and they assemble some way and they interact with one another and they create local biodiversity, and local biodiversity creates some function. My question is, when does this result in that? And so we draw inspiration from the pitcher plants because they're so beautiful, and we imagine a pitcher plant that opens and gets bacteria that come into it. These are the consumers, so it's a consumer resource model. If you're familiar with those, the resources come in, we imagine ants coming in, and then out of the growing and dynamics inside, ammonia is produced, which is then available for the plant to use. And we imagine that each bacterium was consuming some resource, there's a variety of them, it's producing some byproducts that maybe other bacteria can use, it's growing, and it's producing some ammonia that then is accumulated for the plant to use. And then we change different parameters to try to figure out what are the important ones. And so we thought about competition, so we modeled different ways in which they can compete. They can either be generalists or specialists or have this gradient. So here you can imagine that each column is a consumer, each row is a resource, and the color is how much they're using it. And then we imagine by products, like we imagine if you have an ant, you have to kind of like break it down for the next person to use, next bacteria. I keep calling bacteria persons, it's fine, they have a role. Somebody else can use it and so on. And then we use some parameters that are regularly used in consumer resource models, like handling time and resource quality. And then we added something that is very bacterial, which is that co-regulation idea, where the functional activity of a bacterium can change depending on who's around, mostly through quorum sensing, and this can be neutral, it can be intraspecific or interspecific. And I'm going to show you a portion of the results. You can also read the rest of the paper, which is really good. It came out last year. And so I'm going to show you 3 scenarios. Each of these is the same plot we've been looking at the whole time, with richness here and ammonia accumulation here, which is the function that matters here. And this is what we found. What I'm going to do with these is show you which ones of these have significant negative BFs, at least in a portion of their data. And what you're seeing is that the black is the kind of average, and the gray dots are all of the data that was generated. In this particular one, we have that a portion of the generalist and a portion of the gradient scenarios have negative relationships, while the specialists do not. Should have a button for that sound. Okay, and I'm going to show you now the rest, because this is only the co-regulation scenario of interspecific relationships. So here we can say that at least the niche relationship matters, right? Okay. This is a lot, but I'm going to show you stars and what is the name of that thing? Frost, I don't know. And just so you know, so this is flat resource quality, all the resources are the same, and this is increasing. Some resources are more nutritious than others. And these are the, oh sorry, where is it? These are the, I didn't want to show you that yet. These are the co-regulation scenarios. No co-regulation, neutral, interspecific. So what we saw before was this. Okay, ready? Blank. What you're seeing here is one, negative biodiversity ecosystem function relationships are rare across the landscape of possible assembly scenarios, at least for this, the way that we set this up. 2, that co-regulation matters, co-regulation matters, niche relationship matter, and resource quality matter, because these two are different, these are different, and these are different. These are the three parameters that can result in negative biodiversity ecosystem function relationships. So just going back to this, we use these mathematical models to pinpoint what conditions lead to negative biodiversity ecosystem function relationship, and it was these three. This had no role, which I was really surprised. And so to just bring it back to the rest of the story, we use mathematics to again show that at least in the landscape of possible ways in which things can assemble, negative biodiversity is still rare. It doesn't matter, like we don't know how these scenarios are distributed. I don't know if they're evenly distributing across the world, probably not. But even if they were, that would explain why it's so rare and why. And maybe we just really did get so lucky to finding these few examples where we clearly can see it happening in fields, in lab scenarios. So I'm gonna end here, and this slide pretends or tries to make you think about what comes next, like for you, not for me. I know what comes for me. I know my future, but what does it mean for negative BEFs in the things that you're doing? Hopefully I've demonstrated to you that these happen in the field, potentially driven by those negative selection effects, that we can reproduce this in the lab and find out whether there's historical contingency or other parameters that might be affecting the way and the shape of biodiversity ecosystem function relationships, and that we can use math to identify mechanistically how to recreate these. But importantly, we're using richness and diversity here as really an indicator of how species are interacting with one another in the environment and how that leads to the production of some function. And perhaps what I think is we can use negative BEFs to fully and better understand these relationships. And so what I want you to do is think about your systems and how, from the conversations today, I know that a lot of you can apply this to what you do, and how not only negative, but the variety of different ways in which biodiversity and community composition can lead to function, to better try to understand our systems, and perhaps one day, I don't know, conserve some abalone and some seagrasses and some pitcher plants. I want to acknowledge my collaborators for this particular project, Tom, Casey, Bob Gearhart, and the Montreal workshop team, and then people in my lab, the grad students. Everybody's had to do some pitcher plant work because I make them. And then the staff is fantastic too, and about 50 undergraduates have participated in this project, mostly directly in some form. And then all the institutions that have given me an opportunity, I really appreciate it because it's been fun. People that have paid for me to do this work, it just amazes me that people would give you money to play with bugs and come up with strange frameworks. And then I want to acknowledge our staff, like the admin people and the staff that clean our buildings and the people that keep the labs working and the orders coming. This wouldn't be possible without them. They solve so many of my problems every day. And thank you so much for inviting me and coming out today and listening to my stories. And I think we have time for questions. And I'd love to hear from students first, if possible. I warned them this was going to happen. Just momentarily.
Yeah, questions, either on Zoom or in person. Okay.
Oh yeah, Zoom, I totally
We have
forgot.
Zoom. Serena
Hi,
will let
Zoom.
us know when there are some
Okay.
Zoom questions, but
Excellent.
hang on.
Can't perform for everyone, sorry.
That was really cool. Thank you. I feel like I learned a lot about pitcher plants. I'm just really interested in how things get where they are. So like the regional species pool is really cool to think about, but like do you think that two pitcher plants next to each other have more similar communities than further away? Like how are things getting in these and how far are they traveling? And it was cool too that like the two species have different things that are causing high function. I don't know, just I'm interested in sort of like how things are getting in there.
I think we're in the same wavelength. Since I started working with these, my question has been always, how do they get there and how do they move? And the answers are really hard to get. And so I'm gonna tell you kind of like the short version of what I've found out so far. The bacteria seem to be well distributed within a field. It doesn't seem to matter whether leaves are close to one another or even within the same rosette. That doesn't determine bacterial composition, but it does for the protozoa. So we did some experiments where we would seed the protozoans and then track how they would move. And especially the larger protozoans have a hard time moving across the landscape. So there is dispersal limitation that is different across the taxa in the food web, which is interesting by itself. The bacteria, probably not so much. If you look at the community composition of the bacteria within the picture, there's a lot of taxa that are from like soil, characteristic of soil, characteristic of water, like just aquatic communities, and some that could be tracked to insect guts. So probably what's happening is that most of these communities are taxa that live in waters and soils around it, and taxa that are coming with the insects in some combination. We're coming up with some strategies to try to figure out what combination and which ones of these do better in the conditions within the leaf, but it's super tricky because plants are tricky and there might be some coming through the roots, I don't know. So great question, very difficult to answer, and if you have ideas of, we can't tether there like you do with your isoblossom, unfortunately.
This is such a cool talk. Thank you so much. My question, I actually have two questions. The first one is kind of related to Claire's, which is I'm wondering if the pitcher plants themselves do anything to regulate or encourage the colonization by microbes. And then my second question is, you talked about two different species and two different genuses that use predominantly microbes to gain access to nitrogen. And I'm wondering about like these pitcher plants more broadly, and is this like, this isn't the ancestral trait, it seems like, but kind of evolutionarily, what happened so that these two species use microbes and others tend to use more enzyme-like pathways?
Great questions. So let's see if I remember your first one. Your first one was how the plant, does it do anything? So two parts answer. One is when you take the same fluid and put it in a tube and in the plant, they change. They're different. They're like, there's something that the plant is doing, but we don't know what it is, especially for these. I think that experiment was in Flava, in fact. I think it was in Flava. So it's doing something, but it's very unclear what it is. There's one paper that suggests it's pumping oxygen into the fluid, but it was an okay paper. Somebody should follow up. And so we don't know. But Nepenthes, for example, changes the pH. So we can control, we don't know if it's because it's it's the purpose of controlling the microbes, but it can definitely control the pH inside of the leaf. To your second question, what is the ancestral status? So I just submitted a grant to try to get some money to do some of those stuff, but the short answer is that these two species are sister species, along with the sun pitcher, which is the Heliamphora that lives in South America. Heliamphora has been very, very understudied, mostly because it grows on top of the tepuis in Venezuela, where you can't go. And so we don't know what that one's doing. But the Sarracenias are varied, right? So within the Sarracenias, there's Flava and Leucophila do have their own enzymes. It's only Purpurea, as long as I know, that has been tested and doesn't seem to have any. So I don't know exactly what, without knowing Heliamphora, it would be really hard. And if you compare it to the other pitcher plants, there's an Australian one, and then there's the Nepenthes. Those do seem to have their own enzymes. So it's what my idea was for that grant is thinking about broadly carnivorous species. What is their relationship with bacteria in general, and how can we track that to their functions of carnivory, taking into account their structure, so whether they're pitcher plants or sticky plants or snappy plants, and their glands. Their glands are, there's different morphologies of glands, and those seem to be a big important part of going through protocarnivory into the carnivorous mode. So it's a really big question that doesn't seem to be fully addressed, and if you have ideas, I would love to hear them because it's super cool. Yeah.
Hi, thanks for your talk. I have a methods question regarding your in-lab portion. I was curious what motivated you and your team to select the QIAGEN D&EASY kit as opposed to a Zymo kit. Is there a strong difference between the two?
It's a great question. There was not, I think, I'm going back to I think the reason was mostly that when I was in Florida, I was doing extractions with the QIAGEN kit, and then I probably had leftover, and I brought it to Humboldt, and I just used that one. And it also allows me to make really clear comparisons between what I was getting there and what I was getting here. And so most of the decisions in my lab, to be completely honest, about whether to use QIAGEN or Zymo is how comparable we want to make the same story and how much money we have, because the Zymo are cheaper. Being transparent here. But they're both good. Do you have some questions? Nice. No? Okay. Good.
To be a question hog. For your model, I just, like, you found that there were some scenarios that seemed more likely to have negative relationships. Like, do you think that those are actually rare in what you find? Like, seemed like resource quality changed it a lot. So I guess it was like rare among all of the scenarios you did, but do you think that that matches up with what's actually like in pitcher plants and stuff, or does that make sense? I don't know.
Hard question. Hard questions. So you're asking, does this relate to what the system? Okay, I got it. Yes. So, but I'm gonna use this one because I think I got it. We don't know the coagulation pattern. Like, we don't know how these might be co-regulating each other, and we tried to do an experiment with chitin where they were eating chitin, and it was really complicated. So chitin is the wrong one, but we don't know. I would love to know, because I suspect that there's things going on where they're influencing each other, and we don't know for the system. In fact, it's really hard for environmental samples to figure that out, for me at least. Resource quality for sure is different, because it's very different to eat, right when the ant falls in, there's a lot of yummy stuff, and there's like simple sugars and amino acids and things that are easy to consume, versus chitin and like these complicated, lipids and things that take bigger enzymes, and honestly, it takes a lot longer for them to break that down. So this is for sure a reality. In niche relationships, so this implies that there's differences in whether there's hierarchies in competition, and whether that changes from one place to another. It's possible, because it depends on what species are there and how many are there, whether they're gonna end up on this. There are likely generalists in there and specialists in there, and it depends which ones are dominant. It's gonna be more like one or the other scenario. But these are very extreme scenarios, right? We purposely modeled it so it was like the three kind of like very dramatically different ways that it could happen. But yeah, I was surprised that this was not it, because I thought that they would like break down the ant, and then somebody can eat the inside, and then somebody can eat, I was so sure when I was like, run this again, can't be possible. But it like absolutely did not matter what the byproduct system was. And then this one, I haven't seen it in microbial systems as well characterized. But it's so common in resource consumers models that my collaborators were like, we have to include it. And I was like, okay. at all.
All right. Yeah, so we have a question from Zoom. So May says, please correct me if I'm remembering wrong. Thank you for the talk, but could you clarify why the redundant regional micropool was associated with negative rather than neutral or redundant BEF relationships?
Why the redundant was associated with redundant? So this, yeah, is that the?
Why the redundant regional micropool was associated with negative rather than
So
neutral?
I don't know. This is what I expected. And so again, this is very preliminary. I haven't thought about it that much. This is what I expected from this paper. And if you're interested in it, definitely may go and look at this paper because it's really cool. It tries to predict how we can think about the effect of dispersal and regional species pool in the context of function. And what it predicts, it really, honestly, I added this line. It doesn't talk about negative BF at all. It talks about positive and redundant and how it can shift from one to the other. And I just thought, like I put my logic in, and I thought that unique could also mean negative. And why redundant would mean negative, I don't know. I really have to I want to dig into who are these species that are showing up in the low diversity, highly functional, and figure out where they are in the regional species pool and how abundant they are, and try to answer that question. But I don't know, great question. If they have suggestions, definitely reach out because I'm thinking about that.
So I have a question. Are faculty allowed to talk now? Yeah, okay, good. In your very first part, you sort of found these negative associations between diversity and some sort of function, but the explanation was that they're both sort of correlated with successional stage or something like that, right?
In the first one, yeah.
Right. So do you think that is that relationship that you're identifying there, that negative BEF relationship, when you say it like that, it sounds like it's a causal relationship. Do you imagine that to be true, or are those both just byproducts of succession? As succession goes on, other things happen that accelerate function and reduce diversity, or are the diversity and function really actually linked in that example?
That's a great question. I don't have an experimental answer, but we did to kind of tackle that because we can't disentangle it, right? Succession is what created these communities. What we did to kind of get at that is do an SEM and try to see, if succession is affecting function and function is affecting, succession is affecting diversity, diversity is affecting function, and then and it looks like diversity is affecting function, and it's the biggest effect. I think I have a secret slide for that.
That's okay, I trust
I don't remember the numbers, but yeah, we looked at that statistically because there's no way to disentangle it, and that was kind of the reason I started doing it in the lab. So I was like, to really disentangle it, we need to manipulate diversity separate than things that are happening in the field. Otherwise, you're gonna have these tangles, yeah.
Yeah, okay, there you go. I wonder if that's true, then do you think then that the mode of succession, like facilitation, inhibition, tolerance, and so on, that then should affect the relationship between diversity and function, right?
I think so.
So in examples like tolerance sort of models where a bunch of stuff colonizes and things eventually drop out, you get something like what you sort of describe here, but maybe in a facilitation or an inhibition model, you get something a little bit different.
Yes.
Okay.
Yes, and it depends on what the relationship between the facilitator and the functional species would be.
Yeah,
If
and
they're
then-
helping each other, great. If
Yeah.
not,
And then I think that this gets to maybe to Claire's question about which of these things is more common. I think you can think about which of those modes of succession might be most common in different types of systems, and whether that actually drives the direction or shape of a diversity function relationship.
absolutely, that
Yeah.
would be super cool to
Yeah.
do that.
Cool.
Yeah. Succession is great. It's just such a little.
Other questions, either from Zoom? No? Okay. Or anybody here? Well, let's thank Catalina again for a great talk.